Today we're talking about the tools of biotechnology. You can find this information in Chapter 9, Section 1 of your textbook. And before we begin, let's just review what biotechnology is. You've probably heard of it before. It's really a very diverse field, and in biotechnology, we use living things or molecules from living things in a way that makes our lives better in some way. So 3 areas that we'll focus on will be forensics, medicine, and agriculture. Now, before we get into how biotechnology is used, we need to learn about the tools that scientists need in order to actually do biotechnology. And one of the biggest tools that is common in all biotech labs is the micropipette, which you see pictured here. It's called a micropipette not because the pipette is small, but because it can very accurately measure small volumes. And we're talking about on the order of microliters. If you don't remember, micro, this little symbol here, it looks like a U with a tail. That is the symbol for micro. 1 million microliters makes up 1 liter. So a microliter is 1 millionth of 1 liter. So that is a very, very tiny volume, and it requires a special piece of equipment in order to measure something that small. So let's take a look at the parts of the pipette. You have a place to write this on your Power Notes sheet. You have this picture of the pipette here. We're going to start at the top. This is the plunger. That's the part that you push down on with your thumb. That's how you actually control the pipette. Next, we have this little button right behind the plunger. It's called the tip eject button, and that does exactly what you would expect. When you press that button, you can eject the tip, and I'll show you what that looks like on a real pipette in just a moment. Just beneath the tip eject button, there is a black knob. That is the volume adjustment knob. You can see it in this view of the pipette right here. This is the knob that you turn to change the volume. Don't just crank the plunger. Yes, the plunger will turn, but it's not equipped to handle the torque of all of that turning. So really, whenever you change the volume, please make sure you use this black knob right here. Beneath that, there is a window, and this window shows 3 digits. Those 3 digits tell you what volume your pipette is set for. And this is an actual photo of what that would look like down here in the bottom corner. All pipettes, no matter what their volume range, they are going to just show you those 3 digits. So we will talk about how to interpret those 3 digits in just a moment. Next, we have this tip eject shaft here. This on the picture is just an external piece. Looks like it's probably metal on this pipette that would push the tip off the end. The pipettes that we have in class have a plastic eject shaft, and it basically forms a ring around this entire barrel of the pipette down here. And then finally, at the bottom of the pipette, there's a place to attach the tip. All pipettes use disposable plastic tips because you don't want any liquid to go inside the pipette itself. That would really mess up the machinery, which, remember, is very fine-tuned for small volumes. So we always use a disposable plastic tip. It attaches here at the bottom, and when you're done with that tip, that's what that tip eject button is for. You can push the button, it'll push the tip right off into whatever your waste container is that you're using. Okay, so another thing about the pipette is if you look at the top of the top of your plunger tells you the max volume of the pipette. This is a H5000, so that means it can go up to 5,000 microliters, which is the same as 5 milliliters. You can see the range is between 1 and 5 milliliters of that pipette. And here's that tip eject button right here. So let's take a look at an actual pipette and see some of these things in action. Okay, let's take a look at this actual pipette that we have here. Here is our pipette. You can see we have plunger at the top. Here's the tip eject button. There's that volume adjustment knob. Here is our volume window. You can see it says 040 right now. And then our tip eject shaft is this white part here. If I push the tip eject button, see how it moves that whole shaft, and that will help eject the tip from the end of the pipette. I've got some pipette tips here in this box. If you open it up, you can see there's just a few tips here in the top of this box for me to use for a demonstration. So for this pipette, it's currently set at 40. If I look at the top of the pipette, it says H100 on the top. So this would be set for 40 microliters, and we'll do some practice with that in just a minute. But here's how we use the pipette. You hold it like this with the tip eject button facing you. That's where you put your thumb when you eject the tip. Wrap your fingers around this top of the barrel and then use your thumb to press down on the plunger. You can adjust this up to the finger guard if that makes it easier, or really just adjust your grip wherever it's most comfortable on that pipette. I normally hold it up here so I can use this guard here as I'm pressing down on the plunger so I don't have to grip it so tight. Okay, now to use it, all I do is I'm going to put my pipette tip on the end. You can kind of tap that a few times to make sure it's secure. And there you can see now I have my pipette tip on. So let me get that box out of the way. Here's our tip. If I am to draw liquid up into this tip, then I can dispense it very easily into my sample, wherever that is. And then if I need to eject the tip, remember we have that tip eject button right here. I press down on that button and the tip comes right off. Now normally we would eject this tip into a waste container, but just for demonstration purposes, I have it right here. So that's the basics of the pipette itself and how you use it. All right, so now let's talk about that volume readout a little bit more. We do have 3 different sizes of pipettes here. They're indicated on the top of the plunger. This is H1000. It will handle between 100 and 1,000 microliters. And it's good to know that 1,000 microliters is the same as 1 milliliter. H100 can handle between 10 and 100 microliters, and H20 can handle between 2 and 20 microliters. So if you were to set the volume on each of these pipettes, you just have these 3 numbers in your volume window. So how you interpret that is going to be a little bit different based on what the max volume of your pipette is. The way that I recommend thinking about this is the top number of the 3 numbers in your volume window always corresponds to the first digit of the max volume of the pipette. So this one has a max volume of 1,000 microliters. So if I were to set it for 1,000, the 1 would go in the first place here, and then I would have 0, 0. So it would actually look like it said 100, but because I know 1,000 is my max volume, that would be set for 1,000 microliters. If I wanted to set it to something in between, these little tick marks here can get me to that 4th digit, the ones place here. So if I look at this window, it says 075. Well, I know the top number corresponds to the thousands place, which means the second number is the hundreds place, and the third number is the tens place. So 075 on a 1,000 microliter pipette would be 750 microliters. Okay, let's look at the second one, an H100. This is the easiest one because 100 has 3 digits and there's 3 numbers in your window. So the first digit is the 100s place, and then we have the tens place, and then we have the ones place. So on this window, 075 would actually correspond to 75 microliters. And then finally on an H20, this is the smallest volume pipette we have. Now the tens place is the first number because 20 is our max volume. So this is our ones place, and now this is the tenths place, that third digit right here. And the tick marks would actually be the hundredths place. So we can accurately measure out to the hundredths of a microliter in this pipette. So 0.75 on an H20 would correspond to 7.5 microliters. Okay, let's do some practice. Think about what you are looking at here. What volume pipette it is. Look at those numbers in that volume window and see if you can determine what volume this is. If you need to think about it, go ahead and pause the video, and when you unpause it, you can see if you're correct. All right, the volume that would be dispensed here is 10 microliters. 10 because this is an H20 pipette. Our max volume is 20 microliters. So our tens place starts here, then our ones place, then our tenths place. This would be 10.0 microliters. If you need some help with that, please ask about it in class. We have lots of pipettes that you can practice with, and you cannot use a pipette correctly if you don't know how to interpret the volume. Our next tool is a little bit different because this is a molecule rather than a piece of equipment. And this molecule is called restriction enzymes. Restriction enzymes are enzymes, and the reaction that they catalyzed is cutting a strand of DNA. So we can kind of use them just like little tiny molecular scissors. And they come from bacteria. They're naturally occurring, and they allow bacteria to chop up DNA from viruses that might infect and kill those bacteria cells. But scientists have used them in order to cut and manipulate DNA at very specific locations. And the way that works is the restriction enzyme actually cuts DNA at a certain sequence. You can see there are 4 different restriction enzymes pictured at the bottom. This first one is called EcoR1, and it recognizes this sequence, GAATTC, and then obviously the complementary sequence on the other side of the strand. And because it always recognizes and cuts in this one place, we can use it to cut DNA anywhere that sequence exists. We have another restriction enzyme here, XBA1. This would cut at a slightly different sequence, TCT-AGA, SPE1, and then PST1. They all have their own unique sequences where they would cut. And we call that a restriction site. Restriction because it's a restriction enzyme, and the site on the DNA where that sequence is where the cut takes place. Now, different restriction enzymes, as we just talked about, have their own unique restriction site, and scientists can use this to learn something about the sequence of DNA. If this DNA sequence is cut with restriction enzyme number 1, well, whatever sequence this enzyme recognizes, maybe it's located here, here, and here. But if we take the same DNA sequence and cut it with a different restriction enzyme, there might be 6 different places where that cut would occur. And eventually what you will see here is we can use restriction enzymes because they are sequence specific to chop up DNA into a very predictable pattern of fragments. So these 3 cuts in a linear piece of DNA would produce 4 different size fragments. This second restriction enzyme here, it cuts in 6 places. Those 6 cuts separate the DNA into 7 different fragments. A little bit later in the next section of this chapter, we will talk about how to separate those fragments from each other in a process called gel electrophoresis. And that process allows us to learn a little bit about the sequence of a DNA sample just by the way that it gets chopped up and separates into different sized fragments. So we'll talk a little bit more about that later. Before we move on from restriction enzymes, let's talk about the way the cut happens. All of the ones that we looked at so far create sticky ends, and that's what you see here on the right. Notice how the cut is staggered, and it leaves these bases, the AATT, that are unpaired, and that makes it sticky. It would stick to other fragments of DNA with those unpaired bases. This is by far the most common type of restriction enzyme that's used in genetic engineering, because normally when you're engineering DNA, you're trying to stick two different sequences together. So if you have those sticky ends on your DNA, it's easier to stick those two pieces together. On the left, we have what we call blunt ends, and that's when the cut just happens straight across. Notice that there's no unpaired nucleotides, so this makes it very difficult if you were trying to join a different DNA sequence to this strand right here. There's nothing for it to grab onto. So this is much less common in terms of genetic engineering, but it would work just fine if your only goal is to chop the DNA up into fragments and do that gel electrophoresis process like we just talked about. The final tool that we're going to talk about today in this presentation is plasmids. Again, this isn't an actual physical piece of equipment, but it's a molecule found inside of a cell. A plasmid is a small ring of DNA that's found in bacteria. It's not a part of the main bacterial DNA. In this bacterial cell, you see all of this jumbled DNA here. Remember, it's not in a nucleus because this is a prokaryote, but this is the main bacterial chromosome. It's one big tangled loop of DNA. This is the information that that cell needs in order to survive. These little tiny circles here are called plasmids, and they exist for the purpose of having just little special genes, like maybe a gene for antibiotic resistance. They're not necessary for the bacteria to survive and to do its normal thing, but sometimes they can give the bacteria an advantage if they have a certain mutation that allows it to survive some kind of environmental condition. So these plasmids here are very commonly used in genetic engineering. Because they're so tiny, it's really easy to insert them into a bacterial cell. And if you have placed a gene on that plasmid that you want this bacteria cell to express, that's a very easy way to get the gene from outside the cell to the inside of the cell. So let's take a look at what that would actually look like. We have the plasmid here is the blue circle, and you can see it says RES. That stands for resistance. So these are resistance genes. It's resistant to antibiotic A, antibiotic B, and antibiotic C. These resistance genes allow the bacteria cell, if the bacteria contains this plasmid, to not die if exposed to those antibiotics. And that's really helpful in genetic engineering because most of the time you want to kill all of the contaminating bacteria that are just floating around in the air, and you only want the bacteria to survive that have your plasmid in it. Having that antibiotic resistance allows those plasmids to survive when they are placed, or your bacteria to survive when the plasmid is placed inside of it. So that's why a plasmid is generally referred to as a vector. A vector is something that can move DNA. So the vector here is that plasmid. You put the gene that you're interested in on the plasmid using restriction enzymes. You can cut it open and insert the gene. And then that plasmid is used as a vector or a way to insert the DNA into that bacterial cell that you see right here. Another common vector that might be used would be something like a virus. Viruses we'll talk about later in the course. A virus is just a piece of genetic material that's surrounded by protein, and it injects that genetic material into the cell. So that's another really good way to get DNA inside of a cell. So those are some of the common tools that we're going to start out with. In the next section of the chapter 9.2, we will discuss further things like PCR and gel electrophoresis. But if you have any questions about this information, please make sure you ask about it in class, and hopefully you found this helpful.